In this paper, our group introduces and validates a method that uses topologically associating domains, or TADs, to prioritize candidate genes that result from GWAS signals. We call this method TAD pathways. We demonstrate that our method points to different candidate genes than nearest gene in EQTL approaches. We introduce the method as an additional gene prioritization tool that can identify different low-hanging fruit as compared to other traditional approaches. Before I describe the method and results, I will discuss some key terms and ideas. GWAS have been applied to over 300 different traits and diseases and have provided many insights into the field of complex disease genetics. However, GWAS has failed in many regards to explain heritability and to assign functional impact to risk variants. One of the primary reasons for this discord is the fact that most signals fall within non-coding regions of the genome, and determining which genes these regions impact is difficult. TADs are defined by regions of the genome that exclusively interact with each other. TADs also have several other interesting properties. For example, they are largely consistent across different cell types. They are often independent of sequence context and are typically demarcated by insulator elements such as CTCF. Therefore, because TADs define regions of tissue-independent exclusive interaction, we use TADs to define our windows of association to search for candidate genes impacted by GWAS signals. We can represent TADs using the following orange triangle. The triangle shows the propensity of the particular regions of linear DNA, shown as a white bar, to interact. There are genes peppered throughout this folding pattern, with an enrichment of genes near TAD boundaries. A GWAS signal may occur within the TAD, but it will usually fall in a non-coding region. Our method combines these principles to prioritize candidate genes. Taking a step back, let's discuss how GWAS signals are usually assigned to genes. Most of the time, GWAS publications simply report the nearest gene to the signal. Other times, genomic windows are constructed in hopes that a rough proximity will wrangle causative genes. This window is typically constructed either arbitrarily or based on linkage disequilibrium. Instead, the basis of our method is that we construct windows based on TAD boundaries. Specifically, TAD pathways consists of the following pipeline. First, we collect all trait or disease-specific GWAS signal TADs. Next, we collect all genes in each of the signal TADs and curate a large list of every gene that could potentially be impacted by a trait or disease's GWAS signal. We call this list a TAD gene set. The next step is to perform a pathway analysis using the TAD gene set as the input and the human genome as background. The output of the pipeline is a list of candidate genes implicated in enriched pathways. We validate TAD pathways using bone mineral density, or BMD, as an example. We test several genes using molecular experiments that are included in the TAD gene set. After the pathway analysis, we find that the most significant pathway is skeletal system development, which forms our candidate gene list. In this list are many bona fide bone genes that are already well studied, such as LRP5. However, many of the genes predicted to be important to this pathway are not well studied, and their direct connections to bone are not readily apparent. We tested two of these genes, ACP2 and DEF1, which are not the nearest gene to the GWAS signal, and hypothesized that they are important for BMD. Our molecular experiments interrogate this hypothesis. Importantly, software to implement a TAD pathways analysis is available under an open source license on GitHub. We include reproducible examples as presented in this paper. Our strategy to validate these candidate genes involved experimental knockdown of a human fetal osteoblast cell line by siRNA, followed by an assessment of the resulting metabolic differentiation and alkaline phosphatase activity of each knockdown. We first demonstrate the efficacy of our siRNA screens using control genes, our two candidate genes, and their corresponding nearest gene to their respective GWAS signal. We demonstrate the action and specificity of siRNA treatment by quantitative PCR. Consider the following plot, in which expression levels of the gene TNAP, encoding alkaline phosphatase, are shown. The y-axis shows expression relative to untreated control group. The x-axis describes treatment with gene-specific siRNAs and other controls. The colored bar indicates the corresponding siRNA for the gene of interest. In this case, treatment with TNAP siRNA. We see that TNAP siRNA reduced TNAP expression. siRNA against ARHGAP1, ACP2, BET1L, and DEF1 also effectively knock down expression of these genes. Next, we tested if the metabolic and proliferative activities are reduced with siRNA treatment using an MTT assay. we observed a significant reduction in metabolic activity when treated with ACP2 siRNA. Additionally, we measured the alkaline phosphatase activity, or ALP, of each gene following siRNA treatment, in which purple indicates high ALP activity. ALP is normally highly expressed in differentiating osteoblasts. We observed that ACP2 siRNA significantly reduced ALP staining and therefore early differentiation. In conclusion, we have shown that TAD pathways can steer researchers toward candidate genes and can capture different genes compared to eQTL and nearest gene approaches. However, our method also has several limitations. It cannot discover new genes that are not annotated to Go terms. Our method would also have a difficult time assigning variants to genes in diseases that impact chromosome looping. We also experimentally confirm a role for ACP2 in osteoblast mechanisms, which warrants follow-up study. ACP2 is the gene predicted by TAD pathways and is not the nearest gene to the significant GWAS signal. We anticipate that future genomic algorithms that incorporate principles of TADs and other three-dimensional genomic data will improve detection performance and help to discover novel disease biology. Thank you for watching.